Good afternoon and welcome to the American Floral Endowment's Grow Pro webinar series. I'm your moderator, Dr. Haramrit Gill. I am a member of AFE's Young Professionals Council and an environmental horticulture advisor at University of California, Agriculture and Natural Resources. I have a master's in horticulture from Punjab Agriculture University, and recently I completed my PhD in horticulture from Texas A&M University. Today's session is on the integrated control of thrips. On behalf of the endowment, I'm excited to be part of AFE's Grow Pro webinars series that features a new topic every month presented by an industry expert. The attendance for the Grow Pro webinar continues to increase, with today's webinar breaking records with over 450 signups. Thank you again for joining us today. The webinars are free to everyone thanks to the generous support of AFE sponsors. The views and opinions expressed in these webinars are those of speakers and do not necessarily reflect the views and positions of AFE. As an independent nonprofit, AFE cannot endorse any specific product or opinion. Today's webinar is sponsored by Beneficial Insectary, Bioworks, and Captiva Prime. Beneficial Insectary is a pioneer of biocontrol industry in the USA and the top producer of beneficial insects and mites in North America. Bioworks help customers in the horticulture and specialty agriculture market successfully deliver crops to the market with biologically based solutions and support. Captiva Prime, a product of Go & Company, enhances pest control by driving pests out of the hiding, increasing insecticide exposure, and boosting the overall efficacy of tank mix solutions. If you'd like to learn more about our sponsors or are interested in becoming a sponsor for a topic, you can find the information on AFE's website at endowment.org/growpro. The link will be pasted in the chat. Today's session was pre-recorded in English by Dr. Rose Buitenhuis. After the presentation, Dr. Buitenhuis will join us for a Q&A. Feel free to submit your questions through the Q&A feature or chat at any time. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. This session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you can access closed captions in other languages. To get us started, I would like to share a bit about our today's expert speaker. Dr. Rose Buitenhuis is Senior Research Scientist, Biological Control and Program Leader of the Biological Crop Protection Program at Vineland Research and Innovation Center. She is responsible for the development and implementation of biological control technologies for the management of arthropod pests, supporting sustainable crop management practices for ornamental and production horticulture. Current projects in greenhouse ornamentals and vegetables address the whole spectrum of IPM, from the plant resistance, environmental factors, and biological control strategies, and how to integrate pest management strategies in the production system. Dr. Buitenhuis, welcome, and thank you for presenting today on the integrated control of thrips.
Okay, so thank you so much for inviting me to give this presentation today. We have a lot of attendees. That's very awesome. Anyway, today I would like to talk to you about integrated control of thrips. My name is Rose Buitenhuis. As was introduced, I'm with the Vineland Research and Innovation Center for already almost 15 years, and I am the director of the Biological Crop Protection Program. So let's get into this. So today I would like to talk to you about integrated control of thrips and also show that it's not only Western flower thrips that we have to be worried about. I'll talk a bit about the identification of the different thrip species, what it means for an IPM program at your operation, and also talk a bit about some new thrip species, including Thrips parvispinus. But first, to give you a little bit of background, where I work in Canada, thrips have always been a very big problem, and were mainly controlled by pesticides, chemical pesticides. However, within 15 years, the Ontario Canadian greenhouse flower industry has actually switched from mainly pesticide-based to basically biocontrol-based pest management. And this is because Canada does not have as many pesticides registered as in other countries like the US. And of course, because Western flower thrips is a thrips that can very rapidly develop resistance, you will start to understand that this is a recipe for disaster. Because when thrips developed resistance to all our available pesticides, Canadian growers had to make biological control work. And as a consequence, thrips have basically kept me employed for more than 20 years. And in that time, me and my colleagues from all over the world have done a lot of research on biocontrol of thrips. And it's really showing, like this graph here, this infographic shows basically what we recommend now as the biocontrol program for thrips. And Ontario flower growers are mainly managing thrips biologically. So about 70% of a grower's biocontrol budget is spent on thrips. Now, if you spend this much money on thrips biocontrol, you have to make it work for control of other pests as well. So some of the chemical pesticides are for other pests and diseases are not compatible with the thrips biocontrol program. So this has pushed the industry towards using biological control for all pests and some diseases. So what we're seeing here in this IPM strategy is a chrysanthemum crop from start here at the left to finish at the right, and all the different stuff that you can do. So if you're working with cuttings, we recommend to dip the cuttings. Basically assume that the thrips are there on the cuttings, and by dipping them, you get rid of most of them. Then already in propagation, you want to start your biocontrol program with, for example, predatory mites. As the crop matures and gets out of mist and spaced, you want to use predatory mites, biopesticides, supporting predators, maybe nematodes. Of course, do mass trapping, because every thrips that is trapped is one less thrips in your crop. Continue that by using sachets after spacing, again, biopesticides, supplement with predators as needed. And then if there is still thrips at the end of the program, you may have to use a final cleanup spray with a pesticide. But this way you avoid using pesticides for most of the crop period, so that the resistance in the thrips population has at least not built up till the point that the pesticide may not work. So just when we thought that we had things figured out, stuff happens, of course. You never have a solution and then it will work forever. What happened is that every 4 years we do a survey of ornamentals growers and greenhouse growers. And thrips are always listed as the most challenging pests and also causing the most crop losses. However, the last time we did the survey, over half of the growers said they were not happy with their thrips IPM program and also experienced more frequent or more severe thrips outbreaks. So something changed, and we were wondering what changed. So we started looking at the pattern of these outbreaks. And since about more than almost 14 years now, we started noticing more frequent thrips outbreaks. Up to the point that some people started to suspect that we are not only dealing with Western flower thrips, but with a different thrip species. However, no one had done any surveys since the mid-80s. Most of the time, we just assumed that the thrips species were Western flower thrips. Of course, they're super small and quite difficult to identify, so that didn't help. However, it is true that the species of thrips can have a big influence on the efficacy of both biocontrol and pesticide program. So in 2016, Sarah Jones-Wessick from the OMAFRA Ministry, Ontario Ministry of Agriculture, started looking in ornamentals at what thrip species were there. And she found that the majority was still Western flower thrips. However, about a third were onion thrips, which was quite unexpected, but would explain kind of these observations of outbreaks. Of course, with this, we were wondering about other crops as well. So we started looking at tomato, cucumber, pepper, eggplant, even strawberry and cannabis. And in this graph, you see that the dark green is still Western flower thrips. So there's still a lot of Western flower thrips present. But in some crops, especially the tomato crops that we looked at and the cannabis crops, a large proportion were onion thrips. And then, of course, there was a really small proportion, the kind of the gray top of the bars, that were different thrip species. Now, you have to note that this was not a systematic survey. So we did not survey all the greenhouses in Ontario. It just shows a snapshot of what may be going on, and that onion thrips definitely were thrip species that showed up in different crops. In addition to onion thrips, which is actually a naturalized species in Canada, we've been finding more exotic thrips popping up in ornamentals and especially in tropical plants. So this includes, for example, Thrips parvispinus, which has been causing a lot of problems in tropicals, but also a potential problem in greenhouse peppers. And it has been observed in many parts of the US and in Canada. But on the right, you see a big list of other thrip species that have been observed since we started looking for thrips, including, like I said, Thrips parvispinus and Thrips setosis at the bottom. The reason for finding these exotic thrips is that the global trade in plant material is only increasing. So thrips can move with the plant material. We also, when we were all isolated because of the pandemics, a lot of us took up being a plant parent. So the tropical foliage really boomed, was booming business. And with that came tropical thrip species. Also, the trend of several countries to deregister the harder pesticides leaves more opportunities for thrips, and especially the resistant thrips, to make a bit of a comeback. And then climate change may expand certain thrip species host range. And finally, of course, if you start looking, you will always find something. And in this case, interesting thrip species. So where do all these thrips come from? Now, of course, like I said, you have the local species, the Western flower thrips, the onion thrips, that exist outside. So they come in through the vents. And Western flower thrips and echinothrips also come in on imported plant material. So what can you do? I mean, you can use mass trapping and screening too close to the entry points where the thrips come in, like vents or doors, or over the crop so that you capture these incoming thrips. Also, if you know when they're coming in, and most of the time this is the summer season, you can ramp up your biocontrol program prior to this peak infestation. And in the graphs here, you can see two examples. So on the left, you see Western flower thrips inside, which is the solid line, and outside in my region, Niagara region. And you see that the captures inside and outside actually basically overlap. So when the thrips are high outside, you will also get more thrips inside. On the right, you see the same, but for onion thrips. And here you see that, again, the onion thrips outside, the dotted line, peaks kind of like July, August. But then inside, you see that they stay over the colder months of the year. And this is probably because we don't see the IPM programs working as well for onion thrips, because they're optimized for Western flower thrips. The other part of where thrips come from are these exotic species that are not locally established, including Thrips parvispinus, Thrips setosus, and these come in on infested plant material. So what can you do? Definitely the cutting dips will help, and you can dip in biopesticides, entomopathogens, and there have been lots of previous presentations about this, so I won't go into detail. I just want to show you the graph at the bottom left, that we did try the dips for Thrips parvispinus, and it has good efficacy, especially the entomopathogens and the mineral oil products. So the other thing that you can do is quarantine new arrivals until you know if they are infected. And of course, have good biosecurity measures. So do not go from older and contaminated crops into your cleaner, younger crops, for example. So the order of entry is very important. And mass trapping, again, will help take down some of these thrips. Now, it's important to know that contrary to the local thrip species coming from outside, here you won't see this peak of infestation. The infestations won't coincide necessarily with seasons. However, to know what different thrift species you have in your crop, you have to be able to identify them. And in the past, it was very difficult because the traditional scientific keys are very technical and require a compound microscope where you have to put the thrip species on a slide with like a cover slip in a mounting media. So it's pretty complicated. So recognizing this, Sarah Jindvisic from the Ontario Ministry of Agriculture and a senior technician, Ashley Summerfield, actually said, why don't we develop a simple key to important thrips of Canadian greenhouses? Because these traditional keys, in addition to being kind of technical, they also often don't include the exotic species found in greenhouses because they're made to identify thrip species outside in the wild. So our key is designed for growers and consultants and only requires a microscope, as you see here on the picture. So you can easily get one of these, even from Amazon, for about $300–400. The key has very easy to understand language and diagrams. It includes all the species that were found in Ontario greenhouse crops, and it includes potential invasive species as well. But because this is a simplified key, it cannot be used for conclusive identification. If you absolutely 100% want to know for sure what thrift species this is, you still have to send it off to an expert. So I included the QR code to the latest edition of the key because we're constantly updating it. However, if you're not located in kind of northern US, Ontario, maybe British Columbia, then this key may not include all the species that you have. So be careful, be cognizant of this, that if you are in the southern US or elsewhere in the world, this key may not be the best for you. We also, in addition to the key, we developed workshops for growers in Ontario to help growers get started with identification of these thrips. And we developed some helpful videos that are posted on the Greenhouse IPM YouTube channel. And note that between Greenhouse and IPM, there's no space that will get you to the page. And these videos include how to do plant washes, which is absolutely the best way to get thrips off the plant material, including the larvae. It's the most effective method also to detect low levels of the pest. The other videos that are there is how to build an aspirator. So if you do plant taps and you want to collect the thrips that you're tapping out for identification, again, this shows you how to build an aspirator instead of having to buy it. And there's more videos to come. So again, when is it really worth identifying thrips? And the answer to this is really like any time that you see something that's out of the ordinary with thrips. So, for example, if your IPM program that previously worked well is no longer working, if there's a sudden outbreak of thrips or unusual thrips damage, not on the foliage instead of on the flowers, or anything that's out of the ordinary. A thrips outbreak in a crop where previously you never had thrips. Maybe you're looking at the sticky cards and you go like, these thrips look weird. And there's quite a few of them present. And also very important, before deciding to use a pesticide, because some thrip species are more resistant to pesticides than others. And we'll come back to this a little bit later. Now, in most cases, except tropicals, most thrips will be Western flower thrips or onion thrips, but it's still worth identifying from time to time so you don't become just complacent and overconfident, like, oh yeah, it's Western flower thrips, because that's what we did for a lot of years. And it turned out to be quite a few of onion thrips. It's best also to really use a key for identification. A common misconception is that the darker thrips are echinothrips or parvospinus, or lighter thrips are onion thrips, for example. And this is not always the case. On this slide, there's a lot of thrips, and they all look different. They have all different colors. But all the thrips on the left are actually Western flower thrips, and all the thrips on the right are onion thrips. And there's no parvospinus and no echinothrips, and there are even the really dark ones. So Western flower thrips and onion thrips range from very pale to brown and even darker. And the darker morphs are more common in the cooler months. It can also depend on the light you use to observe your thrips, the age of the specimen, if it is dried out a bit, and the preservation method, if you have it on sticky cards or if it has been sitting in alcohol for a long time. All this will affect the color of the thrips. So it's not, body color alone is not a reliable identification feature. Now, I'm not going through the key and teach you in 5 minutes how to identify all the different thrip species, but I do want to show you some of the basics here. And I'll do it comparing Western flower thrips on the left and onion thrips on the right. So what you're looking at really is, first of all, these little eye spots between the big eyes. So you have the big eyes that are dark, and then you have these little eye spots, 3 of them in between on their forehead. And in Western flower thrips, they're red, and in onion thrips, they're gray or even not visible. The second thing that you're looking at is this part of the body, kind of like the behind the head is a body part that's called a pronotum. And in Western flower thrips, you see these big, long hairs, quite coarse hairs on the top and on the bottom of the pronotum. While in onion thrips, on the right, you do see a few little hairs, especially if you look under the microscope, but there's no big, coarse hairs or setae on the top of the pronotum, only on the bottom. And then overall, Western flower thrips is pretty hairy. It has these big long hairs, all even on the head, while onion thrips does not have noticeably hairy head. So that's kind of a preview of some of the differences. And if you will be using our key, you will see a lot of these things. Now, one thing with the key that's really important is always start at the beginning of the key. Because that's how the key works. It's a lot of yes and no questions, one after the other. If you start in the middle, it's not going to work because you will have missed several of the very important questions. So always start at the beginning of the key. In the remainder of this presentation, I kind of want to highlight a few of these new Thrips species, especially onion thrips, and then also later on a bit of Parvis pinus. So I call this the return of the onion thrips, because onion thrips used to be the predominant thrips pest in greenhouses before Western flower thrips took over the world. That what happened kind of like in the 80s, 1980s. So it is a global pest of field vegetables, and most research has been done on outdoor crops, not on greenhouse crops. But like I said, we've recently seen it increasing in prevalence in greenhouse crops, especially because the biocontrol has not been working as well as for Western flower thrips, and growers had to go back to spraying insecticides. Now, that's probably not the best method to control thrips, any species, because a lot of thrip species are likely to be insecticide resistant. We know this of Western flower thrips, but we also see it already in onion thrips and even Thrips Parvis pinus. Also, when you import plant material, because of the high pesticide pressure at the propagator or at the origin where the plants come from, these imported thrips may already be resistant. So even the pesticides that we have here may not even work. So also, you should only spray when absolutely needed, and of course, use a good insecticide rotation strategies to manage the resistance. And know that if you spray, this can disrupt the biocontrol for Western flower thrips, whiteflies like Bemisia tabaci, or other resistant pests. So spraying has to be done with all that in mind. It's also important if you spray to monitor before and after to assess the efficacy of the sprays. Did it actually work? And the impact on any of your beneficials that you may be using. So in the graph here is an example of what I mean. In the bottom, you see here, we looked at the number of onion thrips and Western flower thrips, onion thrips in the dotted line and Western flower thrips in the solid line before the spray, 1 week after the spray, and after 2 sprays. And we see that the dotted line, the onion thrips, actually the spray worked for onion thrips. The onion thrips populations went down. However, we also see that it did not work at all for the Western flower thrips. This population just stayed very stable. And in this case, once you stop spraying, because you think like, oh, my onion thrips are under control, the Western flower thrips may actually get out of control because 1, you killed all their bios, potentially, and 2, they are very resistant. So monitoring is very important. So yes, there may be a place for insecticides, but we wanted to know how can we go about adjusting the biocontrol program. And this was a master's thesis of Ashley Summerfield, and actually we are still continuing this project for several years. But in the beginning, Ashley just looked at like, are the biocontrol agents actually working in the lab? If we give them onion thrips or if we give them Western flower thrips, do they like the onion thrips? And she did trials where she just gave them one of the species at the time, so no choice trials. And she did trials where she put both thrip species together and saw what they chose. And in all cases, like predatory mites, for example, like Cucumerus, Swirsky, even Degenerans or Lemonicus, they all ate both Thrips species, no problem. Swirsky even ate more onion thrips than Western flower thrips. In the choice trials, they also Cucumerus and Swirsky showed a preference for onion thrips. So that they definitely can eat onion thrips. We also looked at Aureus. Again, in no choice trials, Aureus even ate more onion thrips, and in choice trials, it ate about the same amounts of Western flower thrips and onion thrips. So really, the potential is there. The biocontrol agents that we have available will eat onion thrips and as much as Western flower thrips. Now that was in the lab, of course, so we wanted to see what actually happens in the greenhouse. And again, we saw, as you see in this graph here, that Swirsky, Lemonicus, our new predatory mite, Anistis baccharum, and Aureus insidiosus all reduced onion thrips populations after 4 weeks compared to the control. And this was done in these cages that you see here, these dome cages, over the course of 4 weeks. So we still don't know exactly why we do not see this good effect in commercial greenhouses. And it may have to do with where we release the bios or how many. We definitely saw that Swirsky did a better job than Cucumerus, which the results I haven't shown here. But we're doing those trials right now. Now, of course, there's also these foliar biopesticides. So again, we tested them in the lab, and both Bovaria and metarhizium-based biopesticides killed onion thrips. In the greenhouse, it was a little bit different. In the greenhouse, we only saw good control of onion thrips with the oil formulations of, for example, Bioceres and Botanigard, as you can see here. The wettable powder and the Velifer actually reduced the thrips a little bit, but there was no statistical difference with the control. So definitely the oil formulations of Bioceres and Botanigard both worked really well. You can also apply biopesticides in the substrate, like, and in this case, we tested Botanigard, nematodes, and 2 predatory predators like Dolosia, predatory bug, and Stratiolelaps, predatory mite. And we saw that the Botanigard drench actually worked, but that, for example, the nematodes, which usually do work for Western flower thrips, did not work for onion thrips in greenhouse trials. And again, in the lab, the nematodes did work, but in the greenhouse, it seemed to not work. In a separate trial, we also tested Lalgard M52, the metarhizium weekly drench, and we increased the predatory mites. And there we saw again that the Lalgard worked really well as the weekly drench, and that if we released the Stratiolelaps every week, again, we could reduce onion thrips. So with all these trials, it's about 4 years now that we've worked on this, and we're getting closer to a complete IPM program against onion thrips. To summarize it, in the lab, onion thrips is killed by all the natural enemies that work against Western flower thrips as well. But we do see some differences where the formulation, like the oil versus wettable powder, or the application methods, maybe in case of the predatory mites, affect the efficacy of the biocontrol products against onion thrips in the greenhouse. And of course, there's this new invasive thrips, Parvis pinus, that will also complicate the IPM program in crops that it can attack. And it seems that Parvis spinus actually not only attacks tropicals and pepper, but it all the plants that we try it on, it actually likes them quite a lot. So Parvis spinus is quite a big threat. And this is why we're starting right now a new project. Unfortunately, it just started, so I can't tell you much about results. But it's funded by the American Floral Endowment. Thank you very much again. And we matched this funding in a federal funding program of Agriculture Canada, the agri-science program, and got a lot of support also from biocontrol companies. In this project, we're going to look at the biology and the behavior of Thrips parvis spinus, because that will really give us good indications of how to control this new Thrips species. We have to know, confirm where it pupates. We have to know its flight patterns, its life cycle. There's a little bit known in the literature already, but it's not necessarily on the crops that we work on. We also have to, are very excited about trying the clean start, applying the clean start principles to Thrips parvis spinus, because it does come in on plant material. So dips and maybe cold storage or exposing it to UV light will help to reduce Thrips parvis spinus on cuttings or other plant material. Then, of course, we have to compare the effectiveness of biopesticides and predators, just like we did for onion thrips. And I just showed you the results. And then we have to put it all together in a biocontrol program. So this will be several years of research, and hopefully over the course of the coming 3 years, I'll be able to update you on how it's going. Now, in the meantime, there are already lots of resources if you want to know about how to control Thrips parvis spinus. A few months ago, actually Sarah Jendrisic, she gave a webinar in this webinar series on Thrips parvis spinus. And this is just a screen capture of a control program that she proposes. So it includes dipping cuttings, using microbial biocontrol in propagation, dipping rooted cuttings again, continuing the biocontrol program, mass trapping, and then delay pesticide applications as much as you can, or do spots treatments or only certain varieties, just to delay the development of resistance. Also do a good rotation program and spray based on damage thresholds. And these are kind of like the main takeaways that she gave in her presentation that you can look up in the archives of the AFE website as well. It's also several resources. Sarah is very active. She has a blog that you can subscribe to on floriculture. And she has a specific page, you can see it right here, Parvis spinus resources, where she collects all the different resources that she can find on Parvis spinus. This also includes kind of a fact sheet about Thrips identification based on Parvis spinus, because we do get a lot of questions like, is this Parvis spinus? Is this Parvis spinus? I saw like a dark Thrips. Do I have Parvis spinus? So again, you start at the top of this fact sheet and you just go through all the questions, and it will give you a good indication if you have Parvis spinus or not. And this one is also available or available soon on the On Floriculture Parvis spinus page. And of course, our colleagues in Florida also have a lot of Parvis spinus resources. This is a screen capture of their website, of the Drs. Ahmed, Alexandra, Cindy, and Lance, and of the Florida Research and Education Center. So with that, I've talked a lot of biocontrol. don't forget that biocontrol is not the only strategy against thrips. You have to combine it with cultural practices and also choose resistant varieties. And that all together will give you a good, effective thrips control. So it's kind of my reminder slide that it's a whole system that you have to work on, including screening, preventing thrips from coming in, growing resistant cultivars and varieties, and then use biocontrol as much as you can. And then if you still have thrips, then you can start thinking about including pesticide in the program. So with that, I would like to thank all the people that have done all the hard work, counted millions of thrips in the different projects, the different funding agencies and companies that have worked with us. A big thank you to the colleagues from the Ontario Ministry of Agriculture, Sarah and Kara, Professor Cynthia Scott-Dupree from the University of Guelph, and Dr. Rose Labbe, who we are working with now also for the thrips identification, and the Parvis Pinus project from Agriculture and Agri-Food Canada. So here's my email address. If you have any questions, I'll be happy to answer them at the end of the webinar. But if you need to, you can always reach me on this email address. So here is also the acknowledgement of the webinar series. Thanks to all the sponsors of the webinar series. And with that, here, the next webinar will be next month. So tune in for that one. And I'll be happy to answer any questions you have about thrips, be it Western flower thrips, onion thrips, Parvis Pinus, or any other thrips. So thank you very much.
Dr. Buitenhuis, thank you so much for the presentation. I would now like to open the session for the Q&A. Our first question is, do you have any experience using the white mass trapping to monitor thrips migration?
Yeah, definitely mass trapping can be used. There's different colors of sticky cards, including white. It's also yellow, blue, and even green, with or without patterns. So there's a lot of options. What works the best actually seems to depend on the situation. It's probably affected by the different light conditions or the greenhouse coverings or anything. So what you basically just have to do some trials yourself to make sure, like, you just put out different colored sticky cards and see what works the best in your case. Even between different hues of yellow or different types of blue, you'll see soon enough which ones will attract most of the thrips.
Thank you. So our next question is, have you done any research to check if the dipping helps to get rid of insecticide residue coming from the stock farms?
That has been a question indeed that has that I've wondered as well. I've seen some studies that seem to indicate that a high pH of a solution actually seems to affect insecticide efficacy. So, but I don't know for sure if you dip in something like soap that has a high pH, if it will actually break down the pesticide residues. It might, but as far as I know, no one has actually tested that.
Mm-hmm. Thank you. And next question is, please try to explain at some point during the webinar how we can avoid two-spotted spider mites breakout when we implement thrip management program. Also, what insecticides would help to break out mites?
Okay, yes. So I'm going to assume that the person who asked the question is talking about a chemical program. What happens if you apply certain pesticides is that you may be actually accidentally killing the natural biocontrol agents that are present that will kill another pest, like spider mites. Also, thrips do eat spider mite eggs, which is, but I wouldn't really recommend them as a biocontrol program. But anyway, yes. So if you know that you will have a resurgence, you may want to include an extra control method for your spider mites. If possible, if it's compatible with what you're spraying, you can use biologicals like Persimilis or Californicus. If you're going a whole biocontrol program, of course, then that's what you would use. So it's a difficult, I can't really tell you like use this product because it depends on the context. And but if you want to chat a little bit more, just give me an email or talk to your crop advisor.
So next question is, could you please share your take on installing traps within the greenhouse? Is it a good control technique or just a way to attract individuals into the greenhouse?
I don't think installing traps in the greenhouse actually causes more thrips to come in. They will come in anyway, especially because they're mostly carried like on the wind. They're pretty poor flyers. So they may be carried towards the greenhouse and then land wherever they can. So installing sticky traps is actually a really good method to catch mass-trapped thrips. I think you can't go wrong with installing traps. So you want to install them at the points where the thrips come in, like vents or doorways or along paths or in between like old and new crops. You also want to install them right just above the canopy, not too high, because that's where most of the thrips will be moving. Definitely once the thrips are on a plant, they don't really fly much throughout the crop. So put the sticky cards where the thrips are actively flying.
Next question is, what is the height thrips can fly?
Yes, so if you look closely at thrips, their wings are basically just a bunch of hairs, which means that they're not really strong flyers. When there's no wind, they can kind of like jump around a bit and fly. So they don't fly very high on their own. However, they can be easily carried on air currents. So if you have air currents going up, then they can reach pretty high, pretty high heights. That's the word. But so it depends. On their own, they don't fly much, but wherever the wind currents go, they may be carried.
Next question, what are your thoughts on using aggregation pheromones with sticky cards?
I've seen it work really well where the trap catches were really augmented multiple times, like several factors of increased by aggregation pheromones. I've also seen it work so. So I think, again, it depends on the setup of your crop, air currents, a lot of different factors, which I don't know exactly what they are. So again, I would just try it. Try a few weeks with the aggregation pheromones and then without and see what the difference is, if it will work in your case.
So next question, what is the best method for attracting outside predatory thrips or other thrips inside a Canadian greenhouse?
I know in the Netherlands, they're doing research on that now. I would say if you want to attract predators, you have to offer them something that they like. So a lot of predators like to have some extra food, like pollen, if they eat pollen, or nectar. So those are kind of the type of plants that you can plant. And you have to make sure that you're not at the same time feeding your pests, because thrips also like pollen. So that's kind of a trade-off that you have to be careful of. And popular plants nowadays is alyssum. and these tiny little white flowers. But yeah, make sure that you're not turning that into a Thrips factory for the sake of attracting a few predators.
Next question is, what insecticide would work the best for WFT?
Again, it depends where you are, what the insecticides are that you have available. It depends on the history of what the thrips have been exposed to previously. So if they have developed any resistance. So that's a very difficult question to answer without any further information, I'm sorry.
So next question is, could you comment on CO2 saturation as a potential curative treatment?
Yes, in principle, it works. However, if you want to do it on a crop, I don't think you really need a closed environment where you can have high CO2 levels for prolonged stretches of time, which is very difficult to do. So maybe as a cutting treatment or a plant treatment for individual plants, but I don't think they have developed the technology yet to really make it work, especially because you need to balance kind of killing the thrips with killing the plants. The plants also need some oxygen and everything to survive. So you may be killing the plants as well.
So next question is, how effective is the mixture of Botanigard and no-fly for thrips?
That's a good question. I mean, both will kill thrips. I don't think that they enhance each other in a way. So their effects may be just additive. Yeah, that's I don't know. I don't know of any studies where they have combined both. I think at best the results will be additive.
Next question is, will the predatory mites work just as well in the field? Also, will this webinar be available for replay? Yes, it will be.
Yeah, so I think there's a few links in the chat already to where it will be available. Will predatory mites work in the field? For sure, depending on temperatures and humidity. So they're predators in their natural range. They are outside. So there's certainly cases where it will work.
How safe is the LAL guard for use?
How safe is LAL guard for use? Pretty safe. If not, it wouldn't be registered. I don't know what exactly the motivation behind this question is, so hard to say.
Since WFT are more likely to be in flowers than OT, have any trials been run using predatory mites with and without flowering plants for both Thrips species?
Yeah, definitely. Western flower thrips, when there's flowers, they love to go into the flowers, and you'll really see the damage show up. However, all our trials that we've done on the chrysanthemums were done on non-flowering plants, because for most of the production cycle, the plants are not flowering. It's just at the end, when they're close to being sold, that they have flowers. So yeah, that's really, if you can't make biocontrol work at the beginning or the middle of the crop cycle, then it's not going to work when the flowers are there, because your thrips levels will be already too high. And that's why we're concentrating on the vegetative plants for both thrips species.
Did you try less than weekly drenches of Lalgard M52?
What we've tried mostly is weekly drenches. At some point, drenches are less practical to apply because the canopy closes. So there is a point where you would probably stop doing the drenches. We have not played with the interval between drenches, for example, weekly or bi-weekly. There's just not enough time to try everything, right?
Mhm.
We try to do what is practical for a grower.
Mhm. Next question is, WFT control with Botanigard ES and oil may give better or longer control? I think that's their question. Is it a better or longer control? Does addition of Captiva give better control?
As I understand, Captiva is an adjutant. I don't have experience with Captiva, but it may help make thrips walk more on the foliage, which would mean that they may pick up more spores of the products. So it's an option, but I don't know.
Next question is, what would you use to clean up an empty greenhouse after an outbreak?
If it's possible and you can heat the greenhouse to like 40 degrees Celsius, whatever that is in Fahrenheit, 100 something, for long periods of time, like several hours, that will definitely kill a lot of pests. You can leave it bare, like no plants in there for a while. And then before you put in the new crop, you can put in some trap plants, could be beans, could be a pepper plant or anything, just to see if there's residual thrips. Definitely do a good clean out, remove all the weeds if there's any, so that you really don't give the thrips anything to eat while there's no crop.
Mhm. How often would you recommend applications of bioinsecticides like Botanigard?
Usually the application interval that's recommended on the label is like 7 to 10 days, which makes sense if you look at the life cycle of the thrips, because you want to spray when there's new thrips. Basically, there's some like thrips pupae that are in the soil or thrips eggs that are in the foliage, inserted in the foliage, that are not targeted by the sprays. So once those are hatched or emerged, you want to hit them again so that you get all the different thrips life stages.
Mhm. And next question is, how do temperature affects different thrip species?
Maybe a little bit, like higher temperatures, thrips life cycles always go faster. Maybe some tropical species are more comfortable at high temperatures than other thrip species that are more temperate. Not exactly sure if there's like a temperature that we can give, but low temperatures as well. We've seen that Thrips spinus actually dies at lower temperatures, while onion thrips and western flower thrips just slow down and are pretty happy. So
We have still quite a few questions left, but we are coming close to the end of the hour. So rest of the questions could be asked again via email, hopefully. So now I would like to close the session. And with that, thank you so much for joining us today for another session of AFE's Growth Pro webinar series. Join us next month for Dr. Bruce Bugby's presentation on greenhouse climate strategies on Tuesday, September 17th at 1 p.m. Eastern Time. You can register at endowment.org/growpro. While there, check out our past webinar recordings, other grower-related resources, and research reports available to you for free, thanks to the industry support. For more resources on tackling thrips, please visit AFE's free Thrips and botrytis research library to access webinars, fact sheets, research articles, and more at endowment.org/TB. The link will be in the chat below. We ask that you please complete the brief survey about today's session, where you can suggest additional topics and help us continue to improve these webinars. Thank you again for joining us, and have a great day.